So good day everyone. For our chapter 6, we are going to talk about the environmental and non-infectious diseases. Environmental diseases are the most important ones in aquaculture because these include low levels or low dissolved oxygen, high ammonia, high nitrite, natural or man-made toxins in the aquatic environment. So for our non-infectious diseases, these are diseases that are caused by adverse environmental conditions, nutritional disorders, and genetic defects. While they can result to sudden death or mortality, they are not contagious. So first, we have our fish diseases associated with physico-chemical properties of water. So under that, we have our gas bubble disease. So gas bubble disease is due to supersaturation of dissolved gases, either nitrogen or oxygen, and this supersaturation may be due to leaks in the pump or valve systems of our hatcheries. Also, dense algal bloom can be a cause due to depletion of oxygen at night and supersaturation during the day. So signs associated with gas bubble disease includes presence of bubbles in the abdominal cavity, the eyes, the skin, the gills, the fins, the mouth, the swim bladder, and within the digestive tract. It also includes exophthalmia or the abnormal protrusion of the eye or the both eyes. So what could be the effects of this disease on our host? So first, we have death. Death is due to embolism in blood and emphysema in tissues. Next, we have our edema and degeneration of the gill lamellae. And also we have bulging of the cornea and abrupt mass mortalities. For the prevention and control of this disease, we need to monitor the DO or the dissolved oxygen and avoid algal blooms because this can cause oxygen depletion, and maintain efficient operation of water lines and pumps, and fourth is sufficient water exchange. So for our next disease, we have our swim bladder stress syndrome or SBSS. This is a malfunction or associated with the malfunction of the swim bladder and also associated with combination of handling, high ambient temperature, high ambient illumination, and dense algal blooms that presumably can cause oxygen depletion at night and supersaturation during the day. So the effects of this disease on our host is hyperinflation of the swim bladder and can cause high positive buoyancy and mass mortalities. The clinical signs involved with this disease shows that affected fish larvae show large bubble of gas in the region anterodorsal to and outside the swim bladder. For the prevention and control, we should always filter our rearing water, regulate algal blooms in hatcheries, and provide strong aeration to maintain the larvae beneath the water surfaces in hatchery tanks. So next is we have asphyxiation or hypoxia. So what causes this are very low levels of DO or dissolved oxygen. Clinical signs shows that affected fishes gather at the water inlets and outlets, and those fishes that have wide gaping mouth swim at the surfaces of the water and they show rapid opercular movement. So for the prevention and control associated with this disease is we need to always monitor the DO levels and provide aeration. Next is we have our salinity. So progressive emaciation, scale loss, and opaque eye lenses in our affected fish is associated with extremely high salinities. So next is we have our alkalosis. Alkalosis happens when the water becomes too basic, wherein the pH increases to a level higher than the fish can tolerate. So clinical signs associated with alkalosis includes corroded skin and gills, and also there is a milky turbidity in the skin. So for the prevention and control of alkalosis is we always need to monitor the pH level and maintain the pH in a range optimal for the species being cultured. So next is we have our acidosis. So acidosis is caused by a drop in the pH, a level too low for the species being cultured. So clinical signs associated with acidosis includes rapid swimming movements and gasping, increased mucus secretion, and death occur very quickly. So those shrimps that are affected by acidosis shows poor growth, low molting frequency, and yellow to orange to brown discoloration of the gill. So what are the effects of acidosis on our hosts? So the normal metabolism is being hindered, resulting in retarded growth and eventually death. So for the prevention and control is we need to always monitor the pH level and also apply lime and flush pond bottom before stocking. So next is we have our sunburn disease. This is due to excessive levels of ultraviolet irradiation from sunlight when fish are stuck in those uncovered raceways under intense sunlight. So signs associated with this disease includes gray focal circular ulcerative lesions on the top of the head, the pectoral, dorsal, and upper tail fins. So what are the effects of this disease on our hosts? So those ulcerative lesions can serve as a portal of entry for the pathogens and may result in secondary infection. So what could be the possibly prevention and control for this disease is that we always need to place sunshades over ponds and fish should be kept away from ultraviolet installation. Next, we will tackle about shrimp diseases associated with physicochemical properties of water. First is we have our muscle necrosis. Muscle necrosis is caused by temperature and salinity shock, low oxygen levels, overcrowding, rough handling, and gill fouling. So for our clinical signs, affected shrimp shows opaque white areas on the abdomen. In figure 6.3, our Penaeus monodon juveniles have necrosis in the abdominal muscles and also blackening on the edges of the uropod followed by erosion and liquid-filled boils at the tip of the uropods in advanced stages. So in figure 6.4, as you can see, there is blackening in the edges of the uropods and we have this appearance in what we call the wood grain appearance in the abdominal muscle in post-larvae. So what could be the effects of this disease on our host? So there is gradual death of cells in the affected areas, leading to erosion, especially on the tail part of the shrimp. So this can serve as a portal of entry for systemic bacterial infection, secondary systemic bacterial infection. So for our prevention and control, we need to reduce the stocking density and improve water quality by daily water change of up to 5 to 10%. Next is we have our bent or cramped tails or the body cramp. So this is associated with handling of shrimp in warm, humid air, much warmer than culture water and mineral imbalance. So clinical signs associated with this disease is we have partial or complete rigid flexure of the tail, or as you can see in our figure 6.5, the tiger shrimp juvenile has a body cramp. So what could be the effects of this on our hosts is that the partially cramped shrimps swim with a hump abdomen and fully cramped shrimps lie on their sides at the pond or the tank bottom, and those healthy shrimps can cannibalize the weak ones. So the prevention and control for this disease is that we need to avoid possible causes. So next disease is we have incomplete molting. So incomplete molting is closely associated with low temperature in the culture water. So for the clinical signs, there is still presence of the old exoskeleton attached to the newly molted larvae, especially in the appendages part of the shrimp. So in the figure 6.6, this is an example of an incomplete molting in carapace of mysis stage larva under the scanning electron microphotograph. So for the effect of incomplete molting on the host is that there is an abnormal swimming movement which could lead to easy predation and mortality. So what is the prevention and control for this disease is that we need to maintain optimum temperature in the rearing water and always use water heater. So for our next disease is we have our black gill disease. So this is due to chemical contaminants, heavy siltation, and ammonia or nitrite in the rearing water. So also high organic load due to residual feed, debris, and fecal matter on the pond bottom, or for example, the black soil. So signs associated with this disease shows that the gills of the affected shrimps show reddish, brownish to black discoloration, and in advanced cases, the gill filaments became totally black. So for our figure 6.8, this is an example of black gill disease in Penaeus monodon due to heavy siltation. So what could be the effects of this disease on our host? So according to histological observations, there is blackening of the gills due to the deposition of melanin at sites of tissue necrosis and heavy hemocyte activity. So what is the prevention and control for this disease is to avoid heavy metal discharges of nearby factories from getting into the rearing facilities, and to remove the black soil by scraping after each harvest and by draining the pond water from the bottom during the culture period. For the next diseases, we have red disease. So red disease in shrimp is associated with high application of lime in the pond that gives it a high initial pH level, also a prolonged exposure to low salinity. So clinical signs include having red short streaks on the gills or the abdominal segments, as you can see in the figure 6.9, and also there is a yellowish to reddish discoloration of the body. Also, increased fluid in the cephalothorax, emitting foul odor. So what are the effects of this disease on our host is that the histopathological observation of the hepatopancreas shows hemocytic infiltration in the spaces between the tubules, and the more advanced lesions are in the form of fibrotic and melanized encapsulation of the necrotic tissues, either in the tubule itself or the sinuses around it. Also, there is yellow to red discoloration in affected shrimps. So for the prevention and control is we need to prepare the pond bottom properly and to reduce the lime and organic matter content inputs. Next is we have asphyxiation or hypoxia. So this is due to reduced dissolved oxygen due to high organic load or algal bloom and subsequent die-off and high temperature. So for our clinical signs, affected shrimp showed surface swimming and sudden mass mortality. So the effects of this disease on our host includes prolonged respiratory distress, which then leads to death, and sublethal levels may cause impairment of metabolism, resulting in growth retardation. So the prevention and control for this disease is to decrease the stocking density, monitor water parameters frequently, and to provide aeration facilities and water pump for water change. So for our last disease, this is the chronic soft shell syndrome or soft shelling. So this is associated with exposure of normal hard-shelled shrimps to pesticides and pesticides. So aquatine at 0.0154 to 1.54 ppm glutathione A at 1.5 to 150 ppb, rotenone at 10 to 50 ppm, and saponin at 100 ppm for 4 days can induce soft shelling in initially hard-shelled stocks. So for its signs, so the shell is thin and persistently soft for several weeks, and the shell surface is often dark, rough, and wrinkled, and also the affected shrimps are initially weak. So the disease must not be confused with the disease of newly molted shrimps, which have clean, smooth, and soft shells that harden within 1 to 2 days. So what are the effects of this disease on the host? So as I've said, the affected shrimps are soft-shelled and they grow slowly and they eventually die. So for the histopath of the shrimps exposed to glutathione A shows that there is a slight hyperplasia of the gill epithelium, delamination of the cell's lining, and general necrosis and degeneration of these tissues. So for the prevention and control is during the pond preparation, we should flush our ponds thoroughly, particularly when pesticides are used, and also maintain pond water and soil of good quality.